Hello, my name is DuCoeur, and I'm going to be presenting the role of PRKCE in gambling disorders. To start off, the main goal of my project was to just figure out whether the gene PRKCE might influence gambling disorders. I also wanted to explore if there's a relationship between a particular SMP and the gambling phenotype, and also to apply evolutionary theories, and then also to determine if my gene might be essential for survival or not. My hypothesis was PRKCE is an essential gene and has effect on gambling disorders. For my methods, I use several different databases and tools. Also, for all these, I use the resource pages in Canvas that Dr. Dome supplied for us. To select my phenotype, gene, and SMP, I used PheGenI, NCBI, and the GWAS database. I then identified the human protein sequence, and for that I just used Wikipedia. I used BLASTP to find the orthologs in all the 14 different species. I aligned the sequences using Eugene, and then we used clustral O just to um, align them. We used the clustral O, and then also used Eugene to build the phylogenetic trees. Then for the molecular clock analysis, I use just Google Sheets and stuff like that to see if the PRKC follows that predictable rate of evolution over time. For my phenotype, I chose gambling disorder because it honestly caught my eye when scrolling through all the phenotypes. And to be honest, I do like to gamble with my friends, even though I probably shouldn't. Besides that, it is a behavioral disorder that's fairly common, but not as heavily studied genetically, like compared to addictions to like substances. And then, according to the Rutgers Addiction Research Center, about 1 to 2% of adults experience a gambling disorder. And then what I thought was really interesting was um, around people my age, they have a pretty crazy 7.1% rate, and that's according to the NCORE outpatient services. And then the method to measure that very commonly is use the timeline followback method, and this image in the bottom right is kind of a chart for that. You pretty much just go back through and see which days say you gambled, or they also use this for like alcohol addictions, and you could just see and kind of count up and how often. For the SMP results, I use the GWAS database. I found an SMP called RS7591351. It had 6 reported associations, and it's located in an intronic region, which means that it's in the non-coding part of the gene. There was no data about it in ClinVar yet, so there's nothing there. For my gene selected, I picked obviously PRKCE. It has 15 exons, and it is located on chromosome 2, band P21, and it's also been mentioned in about 864 PubMed papers, and that goes back to 1992. Uh, it codes for 737 amino acids and is part of the AGC kinase superfamily, which is like a group important for like cell signaling and stuff like that. The alignment results, when I aligned PRKCE sequences from different species, really not many gaps at all. You can see kind of a bigger group here, which is probably around the 300 and 400. and that means there's just not very many insertions or deletions, which means that it's highly conserved evolutionarily, which means it probably has a very important biological role. For the gene tree results, I found that humans, chimps, and macaques clustered very tightly together, which shows very little genetic distance too. Rodents, rabbits, rodents and rabbits are grouped together with pigs and cattle, which are not too far off there. Birds and reptiles like chickens, alligators, and lizards were quite a bit further away, which shows a big genetic difference. But just overall, um, they're like highly conserved across, especially vertebrates, and they're evolving pretty slowly. For the rate test, I use the dog as the outgroup, and then mammals like humans, cats, chimps, mice, pigs, rabbits, and rats all had their p-values greater than 0.05, which means that um, they evolved the PRKCE gene at like a pretty similar rate to dogs. But the alligator, chicken, lizard, and opossum had p-values less than 0.05, which means their PRKCE gene are evolving faster than the dog. For the reconciliation results, I used the RF distance, which was 6 for me, which means that about 73% of the splits are the same between the two trees. Just kind of overall, the PRKCE shows a pretty consistent evolutionary pattern, I would say, across species. And for my molecular clock, my R² value was 1 for the model, which means it is a perfect fit, but the slope of my real data was 0.0167, which was steeper than the molecular clock model. And this shows genetic distance increases faster than expected, I'd say. There were also some scatter around the trend line, which kind of means that the different species evolve PRKCE at slightly different rates. And then in context of the class, I had the third lowest slope in all the genomics classes since 2022, and it's the lowest of this year's class. And my gene is more stable than the faster evolving genes, which would be like the GAPDH, but it's less conserved than like the one on the top, which is the ACTB, which is like the slowest evolving proteins on this list here. So in conclusion, my gene is highly conserved across mammals and reptiles, and it's evolving pretty slowly. My gene tree and RF analysis show that my gene's structure is very stable, like evolutionarily. Why? My molecular clock shows a pretty clear pattern of slow evolution, and my hypothesis was not supported, but it still is essential, which I got half right, but there's not necessarily a direct link to gambling disorders, so it's still kind of unclear. I would recommend further study because PRKCE plays major roles in the brain signaling, and it's very conserved, which means that it's probably very important in like behavioral processes. Thank you for listening.